Chapter 17, Tumor Immunology. So we'll go over what are tumors, what are the antigens since we're dealing with immunological issues, tumor markers that we actually look for, tests we can look for tumors and their tumor antigens, what the immune system does to help us protect us against tumors, and then how tumors can escape the immune process and immune therapy that's out there. So the first thing is, what is tumor biology? Well, tumor in itself is called a neoplasm. That's just the name for it. That's what you'll hear a lot of the physicians actually call it. And that just means it's abnormal. It can be okay, benign, which means it's not cancerous, or it can be malignant, which is cancerous. So as soon as you hear a malignant tumor, think of it as cancer. So it has a possibility of possibly spreading. Now, what causes cancer? That would be great if we knew everything, but we don't. But they think majority of the cancers are caused by mutations. Now, inside of the actual tumor cell, in the DNA, there's what's called proto-oncogenes. In other words, these are like pre-cancerous tumors, we'll say. And what will happen is something happens to them and turns them on. So there's a mutation in this, and they will turn on and start producing all these cancerous cells. Other things that will happen is normally there are genes in there that suppress the tumors, so they don't do that. They don't become cancerous. So we get mutations in that. So it's like a double hit thing is what they usually call this, is that for the proto-oncogenes to turn on, cancer genes to turn on in the DNA of the cell, or the tumor suppressor genes to turn off. So they called, the brake is now gone and the proto-oncogenes turned on, and means the speed is at its fastest. So it just goes crazy. So with those two things, mutated now, for the proto-oncogenes making oncogenes, which makes cancer-producing cells, tumor suppressor genes being turned off so the tumor can go nuts. So that gives us uncontrolled cell division and a whole bunch of other junk that comes on. So if you look to the picture to the right, you can see in here, these are just looking at some gene mutations in the evolution of cancer. So cells will acquire many mutations over time, and so they get more characteristics, and they learn to evade immunotherapy. So they, one thing is they resist apoptosis, in other words, are immortal, or so they'd have uncontrolled cell division. I mean, they just go crazy. They multiply 10 times faster than normal. Or it can induce angiogenesis. In other words, it can produce its own blood vessels to feed it, which is crazy. And then it can evade or keep the immune system from working on it. So, I mean, it's pretty smart little cookie here. So it knows how to do that. So that's where we're making it very metastatic, so it can invade other cells. So what are the characteristics? Well, we just went through them. They sustain proliferation, they resist death, they induce angiogenesis, they are immortal, they don't die. You mean cells normally die? Well, these don't. And they invade and metastasize. And they avoid tumor suppressors. Remember, that was the second thing we were talking about. So they have an altered type of metabolism. It's a little different. I mean, considering that they resist death and they can produce angiogenesis, I mean, that's pretty remarkable all by itself. And then invade the immune system. So the genes in it, because the proto-oncogenes are now turned on, and or the tumor suppressor genes are turned off, so they have a lot of genomic instability now. So they have an accelerated growth. It just goes nuts. So they cause inflammation on top of it, but the inflammation can't get rid of it. I mean, that's the kicker that comes with it. So certain tumors possess certain antigens, for immunology. That's what we talk about as antigens. So our body can recognize them as foreign, and which is good. So there are 5 different groups of tumor antigens that we have out there. There's mutation-derived tumor antigens. They're called neoantigens, like neoplasm, neoantigens. So they are the proto-oncogenes or the tumor suppressor genes that have been encoded in the cell. And there's, of course, the miscellaneous, the unrelated. So usually these neoantigens are pretty unique to a tumor, and sometimes they can be shared by other tumors of the same type. That can happen. There's mutation-derived tumor antigens, like the BCR-ABL that we talked in CML, P53 and CASP8. We'll talk more about those other, but we've talked about CML. That was one of the first things that we saw. Breakpoint cluster region. ABL is where the Philadelphia chromosome, where we have the flip of 9 and 22. So the cell produces like a tyrosine kinase. It just goes nuts and just keeps, produces tons and tons and tons of cells. So here's what we were just talking about, the chromosome 9 of the BCR-ABL. You see where the chromosome breaks. So a little part of 9 goes on to 12, or 22, and a little part of 22 goes on to 9. So what's happening is we're having massive production of cells, especially in the white cell line, and that's what gives us the chronic myelocytic leukemia. Other types of tumor antigens out there can happen due to viruses that can produce cancers. We call those oncogenic viruses. So they are cancer-causing type of viruses. For instance, Epstein-Barr virus, which causes infectious mono, can actually cause certain B-cell lymphomas, which is pretty scary to think of it. Certain other kinds of cancers can be expressed in certain gametes, sperm and the eggs. And they produce a lot of different types of tumors, like MAGE, MAGE is another big one that we've seen for the those type of cancer antigens expressed in sex cells. There's differentiation antigens that are expressed in certain immature cells of a particular lineage, and we'll talk more about these in a little bit. But CD10, which can be seen on certain B-cell lymphomas, CEA, which is more of having to deal with colon cancer, AFP, alpha-fetoprotein, seen in like liver, and then PSA, prostatic specific antigen, of course, seen in prostate. There can also be overexpressed antigens. They're found in high numbers on malignant cells, and it's called HER2. That's human epidermal region 2 cell line, which is seen in certain malignant breast cancers. So there are some clinically relevant tumor markers. So in other words, there are things we look for in the blood or body fluids or tissues that will give us a key as to what type of cancer it is. So it makes it very specific. So this concentration we look for in the serum will depend on the amount of tumor cell. Given the size of the tumor, like the PSA, if it's extremely high, that will indicate to us, big time prostate cancer carcinoma going on. So elevated levels can indicate a significant tumor mass. So ideally, you want tumor markers to be super sensitive and super specific. Wouldn't that make it nice and lovely? So what they do with these tumor markers is we do screenings. For instance, PSA on men, when they get a certain age, we do PSAs, and hopefully we can pick up that they have early prostatic cancer so that we can identify it. And that's usually seen asymptomatic. So screenings are used to detect cancers at early stages, but you can get false positives and false negatives. If the PSA is very high, they monitor and it stays high, they can do like a prostate biopsy and find out if it's truly cancerous. Like the BRCA genes, BRCA1 and 2, if they're elevated, well, then they might start doing ultrasounds on the breast in addition to the mammography to try to determine if there's cancer there, possibly biopsies, so forth and so on. Because the key to cancer is detecting it early. That's how we can defeat it. So these tumor markers will give us a prognosis as to what's going on. Like the HER2, there are certain kind of medications they can give to that breast cancer that will actually block the estrogen. So if it blocks that, then the the breast cancer can't grow. It will defeat it in a hurry. Works extremely well. So, for instance, the clinical use of tumor markers, here they're using CA, which is carbohydrate antigen 125. You know, a lot of times they can see that marker in certain types of cancer. CA 125 is used a lot of times for ovarian cancer. It can be used for others, but it seems like ovarian seems to be the biggie for this. But anyway, so here's a person, per se. They did surgery, and evidently they had ovarian cancer. All right. She did surgery, so there's decline of the marker. Boom, 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 boom. No evidence of the disease. So they keep monitoring it. I mean, so once you've had this cancer, they'll keep drawing blood, and you can see they're doing it every month. So we can monitor what's going on, because they're floating around in the bloodstream. All right. So here, you know, through April, May, June, and then July, August, you can see there's a relapse. So they'll see, you know, oh my God, the CA-125 is climbing, the ovarian cancer is starting to increase, so they'll give chemotherapy. The CA-125 is still increasing, so that means the therapy is not successful. So they're like, okay, that's not working, let's switch to something else. So they give chemotherapy number 2, and boom, 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 you see the CA-125 go back down again. So it's like once somebody's had cancer, you know, if you're fortunate enough to have a tumor marker antigen out there, you can actually follow it and see what's going on. So it's like a forever kind of thing. And this stuff is constantly being found. There's constantly tumor markers being found, better treatments all the time. That's why it's very important to be very proactive in cancer. So here's some common tumor markers. And you can see it's got the little S, D, and P that's after it. So for instance, the tumor marker AFP, which is alpha-fetoprotein, can be for liver and testicular. And then you see the S, D, P, M next to it. And that stands for screening, trying to remember, diagnosis, prognosis, and monitoring. So that gives you different things that they do for that, which that's not a biggie to me. What's important to know is the marker and the cancer associated with it. So like CA-125 we were just talking about is ovarian. CA-19-9, that's carbohydrate antigen, that's what the CA stands for. CA-19-9 is pancreatic. CEA is carcinogenic embryonic antigen, and you don't need to know what it all stands for. It's colorectal, breast, and lung, but it's big colon here. HCG is testicular, and PSA is prostate-specific antigen. So you can see that they're used for a lot of things. And if you look in your book on page 323 through 325, you can see the CD markers. Excuse me, not tumor markers rather, in there, and the different things that they're used for. And it's important to know these, because yes, I will ask the marker and the cancer that it's used for. That's all I'm after, is those two things. Because we do this in the lab a lot of times. I mean, just look at it. Like we already talked about alpha-fetoprotein. The beta-2 microglobulin, that is big in B lymphocyte malignancies. Because remember, the beta-2 microglobulin, it was a big part of MHC1. So they will see if there's a lot of beta-2 microglobulin, then we're having a tremendous amount of B cell production. So that's an indication of the lymphoma that's possibly going on. You know, so they can sort of monitor, you know, how the B cells are being produced. Calcitonin, more of a thyroid kind of stuff. The CD markers, you know, that's all the white cell stuff. We're going to talk about CA-125. 15-3 is breast cancer. We're going to talk about pancreatic, ER, PR, which is estrogen and progesterone receptors for breast and adenocarcinoma, HER2, which is a breast one. Then the monoclonal, the light change, is more of plasma cells. And then PSA, parathyroid hormone, and thyroglobulin, TG. So you can see there's a bunch of them that we have out there. And this is just a few. There is a lot more that are out there. So it's important to know, you know, the specific ones that'll help us with that. So like I said, the marker and the cancer that is actually used for it. Okay. And it'll explain a little bit more in your book about this if you would like to read a little more as to what it is. So what kind of tests do we do for this? Well, the test that we have for tumors is there's a few ways we can do that. There's immunohistochemistry. In other words, they get the tissues from like the surgeries, all right, the biopsies, and then we can actually get labeled antibodies to attach to the tumor antigens. It's like an indirect or direct fluorescent antibody technique. Cause, so that helps. We can do immunoassays, which is done on a lot of the instrumentation in the lab, so we can look for the circulating tumor markers that are out there. And then there's molecular methods, and we've already talked about a lot of those already. You can use PCR to identify, or you can do cytogenetics, karyotyping, FISH, sequencing of the DNA, or possibly microarrays. Another big one that's out there is called proteomics. Proteomics is looking at the protein profile of the particular tumor antigen that's out there. So proteomics, they can use a mass spec sometimes to identify that, and they can do what's called antibody arrays. That's one of the big ones that we have now. That's sort of like a microarray kind of setup. So it sort of gives us a biomarker profiling the type of tumor that's going on, so we can look at a massive amount in a quick amount of time. So that works extremely well. So immunoassays, that's what a good chunk of the lab does. They're automated, very sensitive, but you have to be careful. Like we used to be doing like PSAs, for example, and if you switch manufacturers, then you might get a different result. That's why it's important to know, when a PSA is being monitored on a patient, that it's done by the same method each time, because if you don't use the same method, you might get different results. And say if somebody was done with method A and the PSA was 5, and then you did that same sample and did it with method B and it was 10. You think, oh my God, it's increasing. Well, because of that reagent, it might be in that manufacturer, it might be 10, might be okay. So that's why it's important that it's the same reagent that's used each time. When we used to have to switch reagents, different manufacturers, we would actually run them in, can't think of the word, together. Can't think of the word here, but they ran them together, and so that the physician could compare, and we would do that for a little while before we totally switched over. So that's one problem. Other things is you can't get false positives. That's why, diagnosis cannot be made alone on just tumor markers. It can't be done that way. That's not giving you a good answer. Other things that we run into is antigen excess, which is called the high-dose hook effect. In the high-dose hook effect, what happens in that particular case is the measurement is now exceeding the linear range. In other words, what has happened is because the growth of the cancer is so big, they produce so many tumor markers out there that in so much excess that the antibody is used up so fast. So it actually eats it up so fast that at the end measurement, it looks like it's nothing. It's like doing a clinic test with a sugar of 2,000. It's like, goes through and then ends up looking negative because it used it up so fast. So that's called a high-dose hook effect. Other things that we run into is interference by heterophile anti-animal kind of things, or autoantibodies. Like a lot of times rheumatoid arthritis will cause a lot of problems with that, giving us false positives. So we have to be careful about stuff like that. Heterophile is another problem that we'll run into. But that's the thing with these tumor antigens. You got to be careful that you're not missing something. A negative might not, might be a false negative, or a positive might be a false positive. So, this is one piece of the puzzle to help the physician make a diagnosis. It's consensual, not the word, repeated times as tumor antigens done is what you're looking at to get us a better clinical picture. All right, so Immune defenses against tumor cells. All right, so there's immunosurveillance. Remember we said we have tumor suppressor genes in there, right? So the immune system will patrol, get rid of any kind of cancer cells, destroys them before, ever causes any kind of problem. So that we have the innate defense mechanism, natural killer cells and macrophages, and the adaptive immune response with the cytotoxic lymphocyte, dendritic cells, cytokines, antibodies. So let's look at a picture here. So here's a tumor cell. Well, it can engulf and kill itself, like a normal cell would do, but God forbid it does that. We can have here the cytotoxic T cell looking at the MHCs. It will produce granzymes and perforins to try to destroy it, or the cytokines coming out from the tumor helper cell. We get the natural killer cell to antigen-dependent cytotoxicity here. The antibody attaches, destroy it that way. It can destroy it by the MHC again that way. If we have antibodies attached, then we have complement to give a membrane attack complex, the MAC thing, because when the tumor cell squirts out antigens, the dendritic antigen presenting cell will then give it to the T helper cell, which will cause cytokines, which is also was over here. But those cytokines will tell the B cells to transform into plasma cells, which will produce antibodies, and the antibodies come attached to the tumor cells, and so forth and so on, which kills a cell. Okay? Isn't that nice? That would be nice if it worked all the time. It's just sometimes these cancer cells just go a little bit crazy. All right? So let's look at this picture here. This is looking at the different phases that tumor cells can escape here. All right. So here is called the elimination phase. Okay, in the elimination phase, this is where the innate and adaptive kills most of the tumor cells, because tumor cells are identified because the MHC is a little bit flaky, so they get apoptosis, blah, blah, blah. Works great. Then we get to the next phase. This is equilibrium. In other words, the immune system controls a limited number, not all, but a limited number of these altered cells. So now the tumor cells begin to lose their identity, that the immune system, that's what it used to identify it as a tumor cell. So it's starting to lose that, or sometimes it can mask it over. So then you get to the escape phase. This is the bad phase. This is where the immune system is now suppressed because the tumor suppressor genes have come out now. So there's chronic inflammation, which means the tumor goes nuts. So the tumor cells are really what they call them undifferentiated. They're just flaky cells. They just grow like crazy. They're resistant to apoptosis. They're resistant to the immune response. They're just going freaking nuts. So what do we do? So one of the things that are out there, and it's pretty big, you see it on TV all the time, is immunotherapy. This is a biological response modifier therapy. This is where we use the ability of the immune system to destroy tumor cells. Yeehaw! So we have active immunotherapy. So in other words, we stimulate the patient's own immune response to the tumor antigens. Now there's some non-specific ones out there. Cooley's toxin, which is a excuse me, it's a real old-fashioned one that's out there. It's not really used that much anymore. And then there's BCG, which is Bacillus calmette-guerin, which was actually used because it's a weakened strain of Mycobacterium bovis. And what it does is they actually get that and they flood the urinary bladder with this stuff, and it induces the innate system and a little bit of the adaptive. In other words, it really gets the T cells kicked up so they start working. My mother actually got BCG because she had bladder cancer, and it worked good. I mean, it kept the bladder cancer in check. And then we have cancer vaccines out there. So we have preventative cancer vaccines, human papilloma virus, right? And then hepatitis B, because that causes liver cancer. And then we have therapeutic ones that are out there. Okay, then it talks about that cancer vaccine, that one at the bottom, that Provenge. That was pretty cool because what they do is you get the patient and then they isolate the dendritic cells, which, don't ask me how they do that, some kind of phoresis kind of thing. And then they incubate it with a tumor antigen. So the gene that codes the antigen to the dendritic cell itself. So these antigen-loaded dendritic cells are then given back to the patient. So they're now like super potent antigen-presenting cells. Hoping what will happen is the patient now, because there's these preloaded antigen-presenting cells, will kick the immune system in the butt and get it going. So it was used to treat advanced prostatic cancer. That was one of the first things that's out there, but they're using it for a lot of stuff now. So it's pretty interesting. This chapter is really super interesting to me. So, because there's tons of cancer in my particular family. All right, so that was looking at what we call active immunotherapy. Like getting the immune system to do stuff. And then there's passive immunotherapy. In other words, so we're going to be giving soluble components, pre-made stuff to it. And one of the big ones that we give is a cytokine to boost the immune response and increase the white cell. Like the, can't think of the words all of a sudden. Colony stimulating factor, CSFs, and that's granulocyte monocyte. Neupogen neulasa, that's a big one out there that can get the granulocytic cells to increase. And then we have interleukin-2, which is a big inflammatory thing. Interferon alpha that are used. So it can try to keep the immune system in check. Try to help either boost the immune system, I guess you could say, or sort of suppress it. I mean, there's different ways that we're looking at there. But like the interferon is used for a lot of malignant melanomas that's out there now, CMLs, Kaposi sarcoma. It has a great anti-tumor effect, so, enhances T-cell response, so it does a lot of things. So, that's the biggest thing with cancers is tons of stuff out there. Like there's certain monoclonal antibodies now that will target specific antigens. They look at the surface antigens, they can look at receptors, and then the angiogenesis receptors or factors that they have. There's immune checkpoints throughout. We have antibody drug conjugates that are out there, like immunotoxins that are out there, and bispecific monoclonal antibodies. If you look in your book on page 337, you will see different monoclonal antibodies that are out there. Like, for instance, one of the big ones that we use right now is rituximab, which is a monoclonal antibody against the CD20 antigen, which you probably don't remember, but I talked about that in a lot of lymphomas because it's a B-cell kind of thing. So, no, you don't have to know, all of this. It's just the different targeted therapies that are out there. Because the names keeps changing. It's hard to remember all these names, but you can see all of them have MAB. All right there, and you see all the commercials on TV, I'm sure. But so they all have different targets that they're looking for and how it works. So a multi-type approach to kill at cancer is what helps. All right. So then we have adoptive immunotherapy. So adoptive immunotherapy is where they transfer cells of the immune system. They excise the tumor, they plate the fragmented cells, they culture with interleukin-2. So once it gets the cells out, and you see there's a whole bunch of cells there, they will culture them with interleukin-2. Now, interleukin-2 really kicks into high gear, we'll say, B cells and T cells. So they'll start doing what they're supposed to do in natural killer cells. So so that they do what we want. So the adoptive immunotherapy, for instance, we're talking about you transfer the cells to the immune system. I got a little interrupted, that's why we're starting over here. But so you saw that they took the tumor out, they get the cells, they incubate them with interleukin-2 to like kick them in there. So they're these Lymphocytes are TILs, which is just another name for tumor-infiltrating lymphocytes. So then these cultures, we then screen them, lymphocytes, and see which ones have the best anti-tumor activity. And then they actually grow them in interleukin-2 again, so they really get in high gear. And then they treat the patient with chemo, and then or radiation to deplete, immunosuppressive cells, and then reinfuse these into the patient. We actually had a guy that was a student here that actually went through that. We take him to Hopkins, and we get all this done. And what this is called CART cell therapy, which is chimeric antigen receptor T cell therapy. That's a fancy name for it. So the patient's, white blood cells are collected, and we put T cells there with them, and we genetically give them the information we want to make them super duper T cells to really get the immune system doing what they want, infuse them back into the patient, and hopefully it'll attack the cancer cells. And this particular student that we had here was working really well, did a really good job. So that brings us to the end, which I think is a very interesting chapter, very changing constantly. So we'll do a little bit of a summary here. So, what is a malignant tumor? Why did we get it? Because of environmental factors, a lot of things. So it gives the proto-oncogenes, in other words, genes that are precancerous in the patient get turned on somehow, or the tumor suppressor genes, which are normally on, get turned off. So we get turned on, turned off, all the same thing, two different types of genes. So these tumor cells will possess altered properties. So they're resistant to apoptosis, unregulated proliferation. They invade near and distant, metastasis. The genomic instability, genes are just, going crazy. They're resistant to the immune system. Normally, the immune cells can recognize these altered antigens because these tumor antigens we were talking about are associated with the tumor. So we can get those associated depending on if there's a virus, an oncogenic virus, So these antigens can tell us a lot of information. We just went through a few examples of what they were. So the immune system will destroy the tumor cells normally by the same way, the innate and adaptive immune system. That's what it's doing. It's doing its job. So normally by immunosurveillance, the immune system can destroy it. But tumor cells are like, So they undergo genetic changes. So they change their genes due to these oncogenes now that are turned on. So they change their genes. So that's because of the changing, and it's affecting the immune response. That's called immunoediting. So it will now escape immune defense mechanisms. So the different tumor antigens that we normally look at, we looked at that in that particular slide. And these are a couple of the examples, but like I said, I want you to know the names of all of them and what organ we're talking about. But alpha-fetoprotein, CA19-9, CA125, CEA, HCG, and PSA. So these tumor markers, we use those a lot of times, and they're done on like the Vista, a lot of chemistry analyzers. So they work really well, but we do get false positives, so that's what you got to be careful of due to some kind of cross-reactivity, like RA, rheumatoid arthritis, heterophile antibodies. We can get false negatives because of antigen excess, that hook effect thing that we were talking about. To treat these things, we can do immunotherapy, so the immune system can destroy tumors, which to me seems to be the way to go. So immunotherapy can be activated due to cancer vaccinations. It can be passive by giving them pre-made cytokines or monoclonal antibodies, or it can be adoptive, where they give tumor-infiltrating lymphocytes, TILs, or use CART therapy. Remember we were talking about the CAR-T therapy is the chimeric antigen receptors T cells that are out there. So I mean that's a good thing. I mean there's a lot of different ways to treat it, and there's not one way to treat anything. There's a multitude of ways because the cancer cell can do so much. So there's many different ways we have to treat it, and that's why it has to be constantly monitored and checked to make sure it hasn't evaded whatever we're using to treat. So very proactive is the key for this. So that was tumor immunology. As you can tell, I really like tumor immunology. So, you can look at the charts throughout, study guides in there, review questions, very good. If you have any questions, just let me know. Thanks.